Good afternoon and welcome to our third and final National Myeloma Month seminar. I'm so glad to be here with you all. My name is Kath Bowley and I'm very lucky to be one of the myeloma support nurses with Myeloma Australia based in New South Wales. I'm joined today by my co-host and fellow New South Wales support nurse, Diana Yun. We would like to begin by acknowledging the traditional custodians of country throughout Australia. We are here today in Parramatta on the land of the Darug people. We acknowledge their continuing connections to land, sea and community. We pay our respects to elders past, present and emerging, and we extend that respect to all Aboriginal and Torres Strait Islander people watching with us today. Our theme for National Myeloma Month has been multiple faces of myeloma, recognising the individual and widely varied needs of our myeloma community. We have some wonderful presentations coming up for you. Today's seminar runs for about 2 hours with a short 5-minute break scheduled between the first and second sessions. Working behind the scenes with us today is our colleague Narelle from WA. She's available on the nurse support line, and if you're experiencing any technical difficulties, you can call her on 1800 693 566. Narelle will also be monitoring your questions, which you can submit today via the Q&A box if you scroll down the page, or via the live chat box, which is available by tapping the 3 dots in the right corner of your screen. We will do our best to respond to your questions today, but please include your email so that we can respond that way if required. You can also email any unanswered questions directly via email to nurses@myeloma.org.au. A reminder that today's session will be recorded and available for playback on our website, along with all of the other seminars from this month. We would love to know what you think about the session today and the other sessions. Your feedback is appreciated so that we can tailor future events to meet your needs. You will find an evaluation form at the bottom of this webpage. Without further ado, it is my absolute pleasure to welcome our first presenter, Professor Joy Ho. Professor Joy Ho is Professor Alan Ung Chair in Haematology at the University of Sydney and Head of Department and Director of Research at the Institute of Haematology at the RPA. She's Head of the Myeloma Unit and Clinical Lead Haematology CAR-T program in RPA. She is the founding director of the Sydney Blood Cancer Research Institute, and she has brought multiple clinical trials on novel myeloma therapies, including immunotherapy and CAR-T therapies, to the RPA and Australia. Joy was president of the Haematology Society of Australia and New Zealand in 2013 to 2015. She's foundation member and was deputy chair of the Medical and Scientific Advisory Group of the Australian Myeloma Foundation and member of the International Myeloma Working Group. She was elected to the board of the International Myeloma Society in 2019, and she's served on the Education Committee since 2016. She served on the ALLG Scientific and Executive Committees and chaired the Laboratory Science Committee in 2005 to 2011. In 2017, she was appointed to the Pharmaceutical Benefits Advisory Committee by the Australian Government. For the American Society of Haematology, she is an invited member of the International Members Committee since 2016 and was appointed the Vice Chair in 2021 and Chair of Metrics Subcommittee for International Programs in 2018 to 2021. Her awards received include the Albert Bakey Memorial Medal, Kanematsu Memorial Award, Arnott Fellowship in Cancer Research, New York Academy of Sciences Fellowship, and Oxford Nuffield Medical Fellowship. She's the author of multiple publications on myeloma, lymphoma, cellular immunotherapy, and hemoglobinopathies. We are so grateful that she can be with us today, and she's presenting on T-cell directed therapies. Thank you so much, Professor Ho.
Thank you very much, Kath, for that very kind introduction. I first want to thank Myeloma Australia for inviting me to speak at this webinar. I think it's particularly exciting that once again, Myeloma Australia has continued to advocate for all patients and doctors treating myeloma during this National Myeloma Month, not only to raise awareness, but also to provide education. And on that basis, I'm aware that my audience in this webinar is likely to be a little diverse. And so in catering for that, I hope to explain concepts on a perhaps more basic level, especially for our patients, for which I'd ask any colleagues who are experts to bear with me, while in some situations, I will also discuss some results at a more technical level so that if there are additional questions, please don't hesitate to ask questions after the talk or even offline, as Kath explained to you just now. I've been given the brief of looking at T-cell directed therapies, and the two main categories of T-cell directed therapies I'll talk about are illustrated on this slide. On the left are the CAR T-cells, and on the right are bispecific antibody T-cell engagers, or BiTEs. My agenda is set out here. I first want to give you a rundown on some of the crucial data, many of which have only been published in the last year. I hope that by the time I finish this talk and I have looked at both CAR-Ts and the bispecifics, we may ask which is better. But you may see that this may not be an appropriate question, as they could, for instance, be used one after another. Secondly, what implications will these developments have on different patient groups? The patient with relapsed myeloma being the key populations we are treating at this stage, either early or late relapse, but also those who are newly diagnosed, whether they're eligible for transplants or not, and the effect of age and performance status. And in relation to this, how and when do we use these technologies? Do we save them until patients break through other therapies, or is it better to use them as early as possible or at diagnosis? And importantly, what happens when they fail? So what is a CAR-T? CAR stands for chimeric antigen receptor, and the T cells are one part of the immune system, with B cells being the other part. The CAR approach uses lymphocytes, which are immune cells transfected by gene therapy, with chimeric antigen receptor genes. They're called chimeric antigen receptor. Chimeric because it combines the effector functions or the immune action of T lymphocytes in killing tumor cells, shown on the bottom of this diagram, with the ability of antibodies from the B cells, the other part of the immune system, as the receptor to recognize targets on myeloma cells in a very specific manner, as shown on the top of the slide in the green part of the molecule. The tumor antigen binding function of CAR, as I showed you and in the top part of the molecule, is usually accomplished by a single chain variable fragment containing the heavy chain and light chain variable domains joined by a peptide. This is from the B cell antibody. This is then connected through a hinge region and a transmembrane domain to the co-stimulatory domains and the CD3 zeta signaling or activation domains, this being from the T cells, hence chimeric. As you can see from this diagram, there are different designs. For instance, the co-stimulatory domain can be a molecule called either 41BB or CD28 with different properties. And as you can also see from this slide, there are different generations of the design as well, incorporating different number of co-stimulators. In very simple terms, this slide summarizes the procedure. A patient undergoes leukapheresis to collect the T cells by an apheresis machine from the bloodstream. These cells are then delivered to the manufacturing site, which is mainly in the US and also in Europe. The cells are selected and activated, and then the chimeric antigen receptor, or CAR, that I showed you just now, is inserted by gene therapy using a virus such as lentivirus. The cells are expanded and are then ready for sending back to Australia. The patient undergoes a short course of chemotherapy called lymphodepleting chemotherapy, which is, as it were, to make space for the CAR T cells to expand, and the CAR T cells are then infused like a transfusion back into the patient. There are two main CAR Ts that have been approved in the United States and Europe for myeloma, and they have the names given here: idacaptogene or ida-cel on the left, and ciltacabtagene or cilta-cel on the right. They both target the antigen called BCMA, which stands for B-cell maturation antigen, a molecule that is highly expressed in myeloma cells and important in maintaining homeostasis of plasma cells. A key difference in the two CAR-Ts is that on the left for ida-cel, it targets one molecule of the BCMA, whereas cilta-cel on the right has two binding domains, and we call it biepitopic. Studies of ide-cel are shown on the next few slides. In the registrational study called KarMMa-1, 128 patients had received the therapy with a median of 6 prior lines of therapy, so heavily refractory patients. 35% of them had high-risk cytogenetics, 39% had extramedullary disease, which is a poor risk factor, and more than 90% were triple-class refractory, meaning that they have failed a proteasome inhibitor such as bortezomib or carfilzomib, an immunomodulatory drug such as lenalidomide or pomalidomide, and an anti-CD38 molecule in the form of daratumumab or isatuzumab. And you will see that this is very much the population of patients that we are dealing with in these pivotal or registrational studies for both CAR-Ts and bispecific antibodies. The response rates are shown on the diagram on the left, whereby clearly there was a dose response, with the 450 million cells that I've marked out for you in that circle being the most effective. At this level, the better than CR rate was 39%, with an overall response of 81%. The duration of responses are shown on the top right, and this varies according to the level of response, with medians varying from 4.5 months to 19 months. The median progression-free survival was 8.8 months, and the median overall survival was 19.4 months. On the basis of these results, ide-cel was approved in 2021 by both the FDA and EMA for patients who have received four or more prior lines of therapy. It's, however, very important to explain, especially for those of you who haven't come across CAR-T cells before, the possible side effects. And the two main side effects of interest, which are particularly of concern in immunotherapy, is cytokine release syndrome and neurotoxicity, which I'll be referring to in both the CAR-T and bispecifics. Neurotoxicity referring to brain and nerve toxicity, which is given the name immunoeffector cell-associated neurotoxicity, otherwise known as ICANS. Both of these important side effects can range from very mild to very severe and can be life-threatening. For cytokine release syndrome or CRS, in the mild setting, this can lead to only fever, but in more severe grades, it can lead to low blood pressure, low oxygen levels, needing even intensive care support. As you can see in this trial, CRS of all grades occurred in 84%, but only 5% were severe. Similarly, for neurotoxicity shown here, it can range from mild grades, for instance, patients have some confusion or have difficulty finding words, to a very severe syndrome with full-blown coma and muscle weakness, again needing life support and intensive care. For this trial, you can see neurotoxicity of all grades was 18%, with 3% severe. Hypogammaglobulinemia, which means a lack of normal antibody production, is also a common side effect, occurring here in 21%, the majority mild. Then there are the whole series of cytopenias or low blood cells, which you can see with the rate of 89% severe neutropenia, low white blood cells, 60% severe anemia, and 52% severe thrombocytopenia or low platelets. Pleasingly, a phase 3 trial is, sorry, I'm just being wrong. Pleasingly, a phase 3 trial is now recently published in February for ide-cel for patients with relapsed myeloma who has received 2 to 4 prior regimens previously. They were given ide-cel at the same dose range, and this was compared with one of 5 standard regimens, which you'll see on the right in that brown box. Again, a highly refractory population with almost with 2/3 of them triple-class refractory and 95% refractory to daratumumab. At a median follow-up of 18.6 months, the median progression-free survival was 13.3 months in the ide-cel group, as compared with 4.4 months in the standard regimen group. A response occurred in 71% of patients in the ide-cel group and in 42% in the standard regimen group. Both were highly statistically significant in favor of ide-cel. Adverse events were as expected with CRS and ICANS. So being a phase 3 study, comparison of two arms, this study is notable for showing superiority of CAR-T over standard therapy in this group of patients. Turning to the other CAR-T cell, which is cilta-cel, this being the one with two epitopes of BCMA targeted, the pivotal study or registrational study called CARTITUDE-1 enrolled patients with relapsed myeloma, again with a median of 6 prior lines of therapy, so highly refractory, having received the same three groups of drugs of proteasome inhibitor, immunomodulatory drug, and anti-CD38 therapy, of whom 24% had high-risk cytogenetics, 13% had extramedullary disease, and almost 90% were triple-class refractory. The results shown here are the latest update published just last year, with a median follow-up of approximately 28 months, showing an impressive overall response of almost 98%, as well as an impressive stringent complete response of 82.5%. The progression-free survival and overall survival curves are shown here, and the survivals were not reached at 27 months median follow-up, and the 27-month progression-free and overall survival were 54.9% and 70.4% respectively. While one shouldn't be comparing between trials, and this is a maxim for all trialists, as it's impossible to know that all the factors which contribute to the results are the same between trials and accounted for. But bearing in mind that the median PFS or progression-free survival and overall survival from the KarMMa-1 study that I showed you just now were 8.8 months and 19.4 months respectively, these survivals for CARTITUDE-1 of median not reached at 27 months appear very impressive. It is also important to know that there are historical studies, including one called Mammoth, which looked at survivals of a similar group of patients who were refractory to daratumumab. The overall survival of those studies was between 9 and 13 months. On the basis of these results, cilta-cel was also approved by the FDA and EMA for patients who have received 4 or more prior lines of therapy. As with ide-cel, I will now point out the adverse events of cilta-cel as reported in the pivotal trial, showing a high incidence of cytokine release syndrome of 95%, but only a small number of 5% was severe. While for the neurotoxicity, the overall incidence was approximately 22%, but 12% were severe. It is also noted that in the earlier patients, in the earlier phases of the trial, there was a late onset Parkinsonian type movement disorder. However, further experience in the use of cilta-cel has enabled us to reduce the incidence because it appears to be affected by the amount of tumor present at the time of treatment, such that if bridging chemotherapy could be used to reduce the tumor load before CAR-T cells are infused, this would reduce the incidence of this late onset movement disorder. Again, as you can see on the left-hand side of the slide, there are significant cytopenias, including severe grades, similar to ide-cel, which are managed as per all hematological malignancies. There are ongoing trials with cilta-cel which come under the umbrella of CARTITUDE-2, and I show this slide simply to indicate that there are a number of cohorts of patients who are intensively reviewed, including those with biologically aggressive disease with relapse less than 12 months after the initial frontline therapy. In the interest of time, I will stick to the published data, and cohort C on this slide was just published about a month ago. And I would like to go through it also because it provides important data to inform overall strategies of giving immunotherapy of both CAR-T and bispecifics. These patients in cohort C progress from treatment not only with the 3 usual groups of drugs I've already multiply talked to you about, but also non-cellular anti-BCMA immunotherapy. And these are either antibody drug conjugates called belantamab or bispecific antibodies, which I'll talk about in a moment, many of which target the same antigen of BCMA. These results are helpful, particularly to understand whether a patient who've had other therapies which target BCMA will still respond to CAR-T targeting the same antigen. In this study, 20 patients were enrolled, of whom 13 were exposed to antibody drug conjugates, or ADC, meaning belantamab, and 7 to bispecific antibodies. 16 of them, or 80%, were refractory, so not responding to these previous anti-BCMA therapy, and 6 had received these therapies as their last line of therapy just before the CAR T. At a median follow-up of 11.3 months, the overall response rate was 60%, and 7 out of the 20 or 35%, or rather 7 out of 10 evaluable patients for MRD were MRD negative. So they were not, that minimal residual disease was not present in 70% of evaluable patients. The median duration of response was 11.5 months. So what this trial tells us is that patients who have failed prior anti-BCMA therapy of other types can still respond to CAR T therapy, in this case, cilta-cel. However, this next slide perhaps bears some of the most interesting messages. The investigators in this cohort C trial analyzed what the factors were that were associated with a response in these patients. Again, considering that crucial question, when a patient relapses from non-cellular anti-BCMA therapy, is it still appropriate to use CAR T therapy directed against the same target? In this study, the factors that are associated with response were shorter exposure to previous anti-BCMA treatment and longer time from the last BCMA treatment to cilta-cel infusion. Importantly and encouragingly, most patients in this study who responded to cilta-cel did not respond to the prior anti-BCMA therapy, again meaning that just because the patient did not respond to the bispecific or belantamab previously does not mean that he or she would not respond to CAR T. So summarizing the developments of CAR T, here's some of what has been done more recently and what's coming, with the trials performed in Australia marked by the red asterisk. Our center at Royal Prince Alfred, or RPA, and others in Australia have participated in the CARTITUDE-4 study shown on the left, which is a study to see whether CAR T should be brought earlier in the relapse treatment regime. This was a phase 3 study looking at cilta-cel compared with standard of care in patients with 1 to 3 prior lines of therapy. This will be reported very soon, and I can assure you will be of great interest. CARTITUDE-5 is currently ongoing in our center as well as others, where we are treating newly diagnosed non-transplant eligible patients with either cilta-cel or lenalidomide maintenance following VRD induction. CARTITUDE-6, this is a trial that we hopefully will be activating soon, is the equivalent of CARTITUDE-5, but looking at transplant-eligible patients, comparing cilta-cel with autologous transplant, hoping to answer the question of whether CAR T will one day displace transplant. The trials listed on the right using ide-cel, which has not yet been in Australia to date, but are ongoing elsewhere, include those looking at high-risk newly diagnosed myeloma in conjunction with other drugs, one of this being a GSI or gamma secretase inhibitor, which is aimed at increasing efficacy because by inhibiting this enzyme called gamma secretase, which reduces the amount of BCMA on myeloma cells, it may increase the efficacy of the CAR T. And thirdly, KarMMa-9, which is likely to be the first ide-cel trial in Australia, in which we will be performing in our center at RPA, comparing ide-cel with lenalidomide maintenance versus lenalidomide maintenance in patients with less than CR after autologous transplant. In addition, and importantly, we at RPA in Sydney and the Alfred in Melbourne are right now performing a novel CAR T study using a new CAR T known as PHE885. This is a phase 2 study, and we are still relatively at the beginning. I will now turn to describe a series of bispecific T-cell engagers, or BiTEs. As I previously explained, a bispecific, as I illustrated on the left, simply refers to the fact that the bispecific molecule is specific for 2 antigens, hence its name, which can therefore be used as an engager on the one hand and be specific for the tumor antigen, in this case myeloma, and on the other hand for the T cell, thus bringing them together and activating the T cell to act against the myeloma cell, hence the name bispecific T cell engagers, or BiTEs. It's important to make the distinction between this from the antibody molecule that I've illustrated on the top right, which has a payload or toxin, such as belantamab, which also acts against the same target, BCMA, but killing of the cancer cells is by a different mechanism, being the toxin. And these are known as antibody drug conjugates. And again, different from an antibody molecule shown on the right-hand side in the bottom that directly acts against a myeloma cell antigen, such as CD38, and which then engages innate immune mechanisms of the patient without the bispecific linkage, such as daratumumab or isatuximab. Since this talk is about T-cell therapies, I will concentrate on the BiTEs. There are now, in fact, an increasing number of BiTEs, and I will start with two which would appropriately be considered the most advanced in development, successively having been published in the New England Journal of Medicine last year, and the first already approved by the FDA, and then mention a few others in the interest of time that we've had experience in Australia. The first is teclistamab, shown here, targeting CD3 on the surface of T cells, and again, BCMA on the myeloma, that is the same antigen as the CAR T cells. And this is the one that has been approved by the FDA. Here is the registrational MajesTEC-1 study, which relapsed patients with at least 3 prior lines of therapy, including triple class exposure to the same 3 classes of drugs. Patients receive a weekly subcutaneous injection of teclistamab after receiving step-up doses which reduce the risk of infusion reaction. Among 165 patients, significant proportions of whom were high risk with almost 26% high risk cytogenetics and 17% with 1 or more extramedullary plasmacytoma, the median number of prior lines was 5, with a large proportion being triple class refractory and pentarefractory, and a median follow-up of 14.1 months. The overall response rate was 63%, with 39.4% having a complete response or better, and 26.7% were found to have no minimal residual disease. The median duration of response was 18.4 months, while the median progression-free survival was 11.3 months. The median overall survival was 18.4 months, though with an amount of censoring, which is a statistical effect. It was thought that the results of the overall survival may not be fully mature, but these results were nevertheless all impressive. Common adverse events included infections, which are frequent in 76% of patients, and severe ones, namely grade 3 and 4, shown on this table, of almost 45%. And this is an important thing to note in bispecifics. Cytokine release syndrome occurred in 72% of patients, but the vast majority were mild, whereas neurotoxic events occurred in approximately 15% of patients, including ICANS in 3%, together with the known cytopenias, including severe cytopenias as shown in the table. The second bispecific antibody that's been fully published only in December in New England Journal is teclistamab. And this is firstly interesting as it's directed against a non-BCMA target, being a molecule called GPRC5D, which stands for G protein coupled receptor family C group 5 member D, a real mouthful again. And it's an orphan receptor expressed in myeloma plasma cells. The Monumental-1 study for teclistamab is a phase 1 study which looked at teclistamab administered intravenously or subcutaneously, either weekly or every other week. Given the effectiveness of the subcutaneous regime that is under the skin, and it being the one that proceeded to phase 2 and will proceed further with development and hopefully registration, I will concentrate on those results. This trial again involved patients who were heavily pretreated with a median of 6 prior lines of therapy, again with significant numbers of patients with high-risk cytogenetics and extramedullary plasmacytoma. Patients received teclistamab at a 405 microgram dose level weekly, or twice the dose every second week. The median follow-ups were 11.7 months for the first group and 4.2 months for the second group, noting therefore that the median follow-up is really quite low in the dose given every 2 weeks. And these are represented in the first 2 columns on the diagram on the right, showing the responses. The overall responses were 70% and 64% in the 2 regimes. And as you can see, the very good partial response or better was 57% and 52% respectively. The median duration of response was 10.2 months and 7.8 months, and the rate of MRD or minimal residual disease negativity was very impressive at 69% to a level of what is called 10 to the minus 5, which is the standard level in the 11 patients who were evaluable for MRD. In patients with previous exposure to B-cell maturation antigen-directed bispecific antibodies or CAR-T, noting that, as I told you before, this bispecific acts against a different antigen, there was a reasonable response, though lower at 50%, whereas the response was also lower in patients with extramedullary plasmacytoma of 40 to 45%. Common adverse events were infections again in just under half of the patients, of which only a relatively small number were severe as compared with the other bispecifics, as you will see. The CRS, or cytokine release syndrome, occurred in 77%, but all but one case was mild, and there was a small incidence of neurotoxicity with no severe events. There were the usual cytopenias, but in addition, there were two rather peculiar effects which are likely related to where the antigen GPRC5D is found. And these are skin-related effects with eczema, pruritus or itching, exfoliation, ulceration, which occurred in approximately 70%, of whom 2% were severe, as well as dysgeusia, which stands for altered taste sensation in approximately 60% of patients. I will now turn to cevostamab, of which we are currently doing a trial at RPA, directed against a molecule called FcRH5, which stands for Fc receptor homolog 5, another mouthful. But this is another molecule which is found exclusively in B cells and at a higher level on myeloma cells. Again, this is administered whereby there's a step-up dosing of a weekly dose in the first cycle and then followed by a dose every 3 weeks. This trial is also of interest as it is of a fixed duration trial, whereby patients completed 17 cycles and then stopped therapy. In the original data of Dr. Trudell and colleagues, which I'll show you in this next slide, it was a phase 1 trial presented in 2021, noting that the drug was administered intravenously in the earlier study, earlier patients. The patients were all heavily pretreated, once again, with a median of 6 prior lines of therapy and 84% of patients who were triple class refractory. The overall response, as you can see close to where the arrow is 57%, with a VGPR or better of 1/3 of patients. Importantly, a quarter of the patients had progressed through CAR-T. In ASH last year, so just in December, a few months ago, another abstract was presented to show that cevostamab had a good duration of response, looking at 16 patients who had completed the whole treatment course of 70 cycles of approximately 1 year. 8 or half of the patients maintained a response for more than 6 months after stopping treatment, and 3 of the patients maintained the response at more than 12 months, although 3 had progressive disease. So the interesting aspect here is that it does show a fixed duration of this therapy, as opposed to indefinite therapy, can achieve continuation of response after completion of the treatment. The final bispecific I will mention is back to BCMA, called elnaratamab, with a phase 1 dose escalation study called Magnetism-1. As before, patients has had a large number of prior lines of therapy with a median of 5. So this study had with 91% triple refractory patients and 31% penta-refractory patients, refractory to 5 lines. Adverse events were very similar to those mentioned before, shown in the table on the right and summarized below. Importantly, however, pointing out, like many bispecifics, there was a severe infection rate of approximately 28%, including one patient who had a fatal adenovirus infection. The overall response rate, again, was approximately 64%, with VGPR or better rate of 56%. Whereas for patients who had previously received BCMA therapies, the responses were still there, though lower, of overall response of 54%. In addition, in 13 patients who had CR, all of them were MRD negative. Following this, 3 other studies are ongoing or starting. The phase 2 study of magnetism-3, looking at patients who are triple-class refractory, magnetism-5, comparing alniratamab alone or in combination with daratumumab versus daratumumab, bortezomib, dexamethasone, and magnetism-7, in which we are participating, being a phase 3 study using alniratamab after autologous transplant. In magnetism-3, a cohort of the patients was reported in ASH just a few months ago for the subgroup who had not been exposed to BCMA therapies before. And in the interest of time, I will show this one slide summarizing the overall response, which were comparable to previous, as you can see, approximately 61%, with both median progression-free and overall survivals not reached, but emphasizing again the importance of infection, with a rate of approximately 2/3 of patients with 1/3 being severe. This report also providing data on COVID pneumonia because the trial was conducted during the pandemic, with 1/4 of patients infected and 2 died of them. Here's a summary slide of some of the agents that are available currently on clinical trial, of which, as I said, one of them has already been approved, teclistamab being on the top line. In this fast-changing field, there are actually progressive changes to this table all the time. So if you search on the internet, there are various versions of this table. Overall, you can see, and I've marked out the key messages by the arrows on the top of the table. The first arrow showing that subcutaneous or under the skin dosing is more predominant and appropriately so, that response rates are in the 60 to 70% mark in the second arrow. The third arrow showing you that the total CRS or cytokine release syndrome rate is high, but most of them are mild, and that neurotoxicity rates are overall low. And in the last column, I'm showing that in many trials, there are now data on the response rates in patients who had previously been treated by BCMA-directed agents, some of which I've already shown you, and this will be likely be of increasing significance. I decided to show this older slide, as I've told you, these slides of BSAs are continually evolving, which is a good thing, because it shows the rate of infections as a summary with at least 40 to 50% infection rate, emphasizing the important message that this is a significant issue for bispecifics, and prophylaxis or prevention by antibiotics is increasingly considered. So how does one pick from this list? Obviously, one would look at the efficacy with respect to response rate, durability, and ultimately survival, although the data for survival do take a longer time to become available. And as you could see from my presentation just now, many of them did not yet have informative survival curves, just response rates. The side effects are important, including infections and CRS. One may also choose according to the target, because many CAR-T targets are still directed to BCMA, though not exclusively. It may be possible to try to use non-BCMA targets initially if there was a possibility that a patient may benefit from CAR-T in the future. Even though from the data I showed you just now, there are responses in patients who have been exposed to CAR-T directed at the same target. Of course, cost and availability are crucial issues. Furthermore, there are already combination trials, as I alluded to, with known agents such as anti-CD38 or daratumumab and proteasome inhibitors, and their efficacy in combination may affect the choice as well. Finally, I'd like to look at what to do when a patient fails therapy, and in particular from CAR-T therapy. Despite our hopes for CAR-T, unfortunately, patients still do relapse. And this is a review performed at University of California, San Francisco on 78 patients who relapsed from CAR-T. Here are the therapies that these patients received following relapse, and there's a whole variety of them. Some went on to another CAR-T and other therapies, including bispecifics, and there was, as you can see, still a significant percentage of response in some of these patients. And here you can see the median overall survival following the relapse from CAR-T was 14.8 months, bearing in mind that this is a median level. And of interest, the median overall survival of patients who received another salvaged BCMA CAR-T or bispecific within 6 months of initial relapse had a median progression-free survival of 18 months, which is an encouraging finding and higher than the other figure that I mentioned just now of 14.8 months. So why do patients relapse either from CAR-T therapies or BCMA? This slide explains some of the possible mechanisms of CAR-T resistance, which I will also apply to BCMA as required. The mechanism may be intrinsic to the CAR-T cells, whereby either the cells don't persist or they become exhausted and therefore inactive, as shown in the left-hand panel. Sorry. They may also relate to the myeloma cell, in which the target, whether it be BCMA or something else, is expressed at a lower level, such that the T cell recognition and killing mechanisms therefore become less effective. And this would also apply to the bispecifics. Whereas the final mechanism shown on the right may be due to the microenvironment, which is the milieu in which the myeloma cells thrive, whereby there may be molecules or chemicals which suppress the activity of the CAR T cells or the patient's own T cells in the case of the bispecifics. There are now clear data in the literature showing that native T cells in myeloma patients are deficient in activity. And here are some of our data showing that they have low proliferation and senescence, and that these characteristics occurred in T cell clones. Our colleagues in the in the United States have since derived data that a patient's T cell health, with those who have fewer of those senescent clones and a more diverse repertoire of T cell activity, recognizing more proteins and antigens from the cancer, is associated with longer durability of the CAR T response and longer progression-free survival. Continued efforts are also being made to expand this repertoire of targets of T-cell-directed therapies, as shown in this recently published diagram for both CAR-Ts and BiTEs. So I've actually come to the end of my talk, and I'd like to end it with a number of questions for the future with some of my own answers, but I've put the word answers in inverted commas because this is as far as I can see them, and we certainly don't have all the answers yet. First of all, is it a matter of CAR-Ts or bispecifics or both? And if both, how do we sequence them? Well, we don't know yet. And in a practical way, given the restricted availability in our jurisdictions, the practical advice would be to use them as they become available and according to the data available so far. This will obviously depend on a few key points. As I've shown you, there are certainly data available that CAR-T and BiTEs are effective in some patients with prior T-cell therapies, even if they've been treated before. And this works in both directions. The second point being that we've seen the durability of CAR-T therapies because the follow-up of the main trials of CAR-T has been longer. What we don't know yet is whether the BSAs will match the durability of the CAR-T. And this is why I mentioned the follow-up in each of the trials. As I already mentioned, we do have combination trials occurring. Will this affect efficacy and safety? The next question is, as I just mentioned, myeloma is a disease which associated with problems with T cell function, as data from our own laboratory have consistently shown over many years, and that studies have now shown that T cell health matters. So should we use CAR-T earlier, or should we even consider collecting T cells at an early stage of disease for a rainy day? And I raise this issue with a bit of trepidation because this is a very resource-heavy undertaking to collect for a rainy day. And even in autologous transplant programs for stem cells, it is in general not possible, due to the resource issue, to store cells for the future just in case. Then there is the related and very challenging question that hopefully the current trials will answer. Could earlier use of CAR-T in fact lead to a cure? And related to that, could current modes of therapies, such as autologous transplant, be eventually replaced by CAR-T? And that will be answered by one of the trials that we and others hope to commence in the near future in Australia, CARTITUDE-6. And the final and important question is whether CAR-T should be reserved for late lines or aggressive disease or used earlier. To my mind and others' minds as well, the answer is probably not. From the KarMMa-3 study data that I showed you, and given the effectiveness of cilta-cel as seen in CARTITUDE-1 for late relapse patients, we also eagerly await the answer from CARTITUDE-4 in patients who are treated with cilta-cel with 1 to 3 prior lines of therapy that is earlier in their relapse. I will stop here And I'm happy to take any questions. But before I do that, I'd like to acknowledge all the colleagues in my unit, including the myeloma research unit, especially Dr. Christian Bryant and Edward Abadia, and our clinical trials unit colleagues, and all the patients and families who put their trust in us. I will turn back to the last slide in case it might generate some thinking points and questions. And thank you very much for listening to me.
Thank you, Joy. Such an informative session and encouraging, isn't it, to see all of these drugs in research and trials, especially when you say that slide is changing almost weekly, it would seem. Thank you for covering some of the questions already in your last slide. We do have a few that have come through from the live chat, some in line with what you've mentioned there, particularly the last question there. There's a question just about whether high-risk cytogenetics would make it more or less likely that T-cell directed therapy would work.
This is something that is not known. And as you know, with all clinical trials, it is always important to analyze the high-risk cytogenetic group to see whether that group has affects the overall response. And it would be fair to say that from the results available so far, these patients do respond. And I would answer your question simply by reflecting the data that have been published and indeed in many trials. I would say to you that it makes more sense for the use of CAR T cells because the mechanism of killing is very much across the board. If the antigen is there, and all the surrounding conditions, including the microenvironment, as I said, the expression of the surface antigen, and the CAR T cell being present and not exhausted, it should work. Then you will say, well, why doesn't it work in some high-risk cytogenetic patients? It could be those very same factors. So my answer to you is that it makes sense to use T-cell directed therapy in those patients with high-risk cytogenetics because it may, in a general sense, overcome some of the other mechanisms which lead to resistance with cytotoxic and other chemotherapy.
Thank you. Thank you, Joy. Another question, which we do probably hear a fair bit coming through our groups, given that we look after regional areas as well. But if someone's interstate or regional, how might they get access to these trials, Joy, that do seem to be focused in our major metro hospitals?
Yeah, so be fair to say that in the initial trials that they, and particularly because RPA has been in CAR-T since the initial lymphoma programs. So obviously with that experience, those centers which started doing CAR-Ts, in our case, it was in 2015-16, so it's now 7 years, tend to have that experience and would be more selected, I guess, for subsequent trials. But I want to encourage our patients in all the rural regions to say that from our experience with myeloma, which has really been a second wave because lymphoma and acute lymphoblastic leukemia led the original strategies. So you will see, for instance, that CARTITUDE-4 was more widespread. Certainly CARTITUDE-5, in the case of CARTITUDE-4, there was only Sydney and Melbourne, but CARTITUDE-5 has expanded, and I suspect that CARTITUDE-6 is also very similar to CARTITUDE-5. So I'll answer the question by saying that I anticipate the studies to be kind of more widespread. I do understand that they still have to be in metropolitan hospitals because there is a significant amount of infrastructure that is required in terms of collection of the cells, processing of the cells, as well as the care of the patients with very close communication, for instance, with intensive care, neurology, respiratory physicians, and infectious diseases, given all the possible side effects I've told you about. So it still necessarily needs to be done in metropolitan regions, but I'm sure that those metropolitan regions will expand.
Thank you. Another question coming through from Veronica, who's asking about the likelihood of a therapy being approved in Australia in the near future, and asking whether cilta-cel will be put up to the MSAC again this year. Are you able to comment on that?
Um, I can't comment on whether it will likely be approved in the future, but I am very I'm aware that it will be it will be reassessed. Don't
Thank
think
you.
I think that is well known.
And another question that came through was about the manufacture. So knowing that, people are aware that it's expensive because these things are coming from overseas, is there any moves for local manufacture of CAR T therapies or the bispecifics?
Yeah, so um, at the moment, myeloma CAR Ts are all manufactured overseas, and this is related to the fact that the sponsors or the manufacturers who devised the original myeloma CAR Ts were located overseas. It would be fair to say that there are a number of units which have the capacity to manufacture in Australia. And I will say to you, for lymphoma, currently there is a clinical trial in Brisbane which is in which CAR Ts are manufactured there. So I would say to you, look, there is the hope that there will be local manufacture, but I really can't say anymore, because as you can imagine, it's not only the knowledge or the capacity, but it's also the tricky issues of funding and so
Mm, absolutely. Thank you. We are out of time. Any other questions, we will respond via email. So please do submit them via the nurses@myeloma.org.au email. Thank you so very much, Professor Ho, for your time and your expertise. It's been really wonderful to have you speak today. Thank you so very much.
Thank you very much, Kath and Diana, who
Thanks.
is also on the line. All the best. Bye.
Thank you. And for everybody else, we're going to have a short break now, a 5-minute break. So take the opportunity to grab a refreshment, have a stretch, and we will see you back here in 5 minutes for our second and third speakers. Thanks so much.
Hi everyone, welcome back after a short break. My name is Diana and I'm one of the myeloma support nurses from New South Wales that's also chairing this event. I just wanted to remind everyone that if you have questions, just remember to pop it in our live chat feed so that we can answer them whenever we can. And I just wanted to introduce you to our second speaker for today. So here we have Dr. Hannah Wardle. She is an early career researcher with a vision to eradicate the physical, psychosocial, and economic burden associated with cancer and its treatment. To date, her research has been critical in understanding how the bacteria in our gut contribute to certain side effects of the chemotherapy, and she has published more than 50 research papers in this area. She is also forging a strong international profile and continues to increase her leadership capacity in this area. At the moment, she is the vice chair of the mucositis study group, and she hopes to use this opportunity to improve collaboration between different study groups and multidisciplinary members. A little bit more background history. So Hannah was under a fellowship and was in the Netherlands, where she led a program of research that aimed to develop and test new strategies to better support models in conducting both clinical and industry-supported research projects. And since she has come back to Australia, she's also established her own one called the Supportive Oncology Research Group. And their aim is to have strong foundations with consumer engagement, collaboration, and to make an impact. And they have secured more than 3 million in competitive research funding and currently have a 1.5 million funding to support a new clinical trial to investigate if medical cannabis improves well-being in people with advanced cancer. She also has a great partnership with the BioMe Bank, which is Australia's largest stool bank, and is working to establish a fecal transplantation service for people who have had a stem cell transplant. She is passionate, and she is also a very good advocate. And she has been recognised as one of South Australians 40 under 40s in 2020. And she's also the 2021 South Australian Tall Poppy Recipient of the Year. So without further ado, let me introduce you to Dr. Hannah, and she can give us our second talk.
Thank you very much for that introduction. I always feel a little bit awkward with that introduction because it makes me feel a little fancier than I really am. But I think hopefully through that, what came through is that I am a strong advocate for improving the quality of life of people undergoing cancer therapy. And I have a very strong interest in people with blood cancer, in particular myeloma. I work heavily with the Myeloma Research Laboratory here in South Australia because we know people with myeloma seem to do it particularly tough with some of the physical and psychosocial consequences of their treatment. So for today, we're going to be talking about a topic that is absolutely dear to my heart, and that is the gut microbiome, which is really all the bacteria in people's guts. It also includes other microorganisms, but bacteria certainly dominate our intestinal tract. And a lot of my work is understanding the changes in these bacteria and the broader gastrointestinal environment in myeloma and other cancers, and looking for ways in which we can better support gut health throughout treatment. So as I mentioned, my name is Hannah and I'm a laboratory scientist by training. And so what I really want you to remember throughout today's presentation is that I'm not a clinician. I'm here aiming to provide new insights into the mechanisms of some of these consequences of treatment and hopefully develop some new solutions that with collaboration with our clinical partners, we can get into clinical trial and into clinical practice. So in today's presentation, what I will be covering is firstly, I really want to emphasize that supportive oncology is really an integral part of modern cancer medicine. I know you've had some presentations over the last few sessions or webinars that have focused on symptom management and quality of life. And I think it's really fabulous to see that because that's not always the case. I then will discuss how myeloma drugs negatively impact gut health before moving on to approaches in which we can support gut health in myeloma. Some of these, or most of these, are very much in the experimental and development stage. And finally, of course, because we know that many people with myeloma, particularly people that have received stem cell transplantation, are largely immunocompromised, I will discuss briefly some of the safety considerations for bacterial interventions in myeloma. Okay, so supportive oncology, what is it, and really why should we care? Well, what we know is that cancer survival is really increasing quite rapidly, and this is reflecting some of these incredibly sophisticated new therapies like the CAR T cell therapy we've just heard about. And we now know that the 5-year survival rates for all cancers is above 70%, and for myeloma, it's above 50%. And so, what this means is that we are really faced with a large and growing population of people who are living with or beyond a cancer diagnosis. Now, what we know is that when people are living with or beyond a cancer diagnosis, this doesn't necessarily mean that everything is hunky-dory. We know that a lot of people will experience an unwanted symptom or side effect as a result of their treatment or their disease. And this unfortunately persists chronically in about 30 to 40% of the time. And so I think it really comes back to this concept of not dying does not always mean living. I really want to emphasize that with a cancer diagnosis, there are a range of symptoms and side effects that will persist. And this really does impact on the individual's ability to live a healthy, happy, and fulfilling life. And in the context of myeloma, or more broadly, I should say, supportive oncology really is this umbrella term used to describe the strategies, whether they be pharmacological or non-pharmacological, to prevent and manage these adverse events. Now, as I mentioned, gastrointestinal health or gut health is one of my particular areas of interest. I did my fundamental training in gastrointestinal physiology and immunology. And so I'm really interested in understanding how the drugs we use to treat myeloma impact gut health. Now, we know that nausea and vomiting is a pretty common symptom. However, increasingly, whilst we consider it to be a gastrointestinal symptom, I suppose, we now know that most of the mechanisms that drive nausea and vomiting tend to come from changes in the brain. However, we also know that some of the drugs used to treat myeloma cause bloating and abdominal pain. They can cause diarrhea, and they can also cause constipation. And this tends to be a bit of a silent one. People don't really notice that it's a problem until it becomes a big problem. And so constipation is quite a strong interest of mine. Now, in terms of the sort of trajectory of these symptoms, we tend to see that nausea and vomiting are usually short-lived after chemotherapy and on the whole are relatively well managed. That's not to say they aren't a problem in some people, but typically these symptoms are across the board relatively well managed. Diarrhea is more common after bone marrow transplantation because of the intensity of chemotherapy that is used, but it's also seen in lenalidomide therapy. So when people are receiving lenalidomide as a maintenance therapy, we tend to see quite a lot of diarrhea. And constipation and bloating tend to be more common with Velcade or bortezomib, and I'll explain the mechanisms of this in a little while. So how do these drugs cause these symptoms? Well, in terms of chemotherapy, we know that most chemotherapeutic drugs work by targeting rapidly dividing cells. So cells that are consistently replicating and replacing themselves. And this is a very common or core trait of many cancerous cells, but it unfortunately is a very common feature of many healthy cells. And so the gastrointestinal tract is one of the most rapidly dividing cell populations in the body. We see a total replacement of the lining of the gut in about 24 hours. And so because of this inability of chemotherapy to differentiate between these cells, we see a lot of cell death in healthy cells that are rapidly dividing. So just like those ones throughout the gut. So what we can do is we can look at the microscopic structure or architecture of the intestine. So on the left-hand side here, there's an image of a cross-section of the intestine. And one of the most important features of the intestine is these really long finger-like projections. These are called villi. And after chemotherapy, this is after a drug called melphalan, which is commonly used in stem cell transplantation, we can see that these long finger-like projections become very blunt and the general architecture of the intestine becomes very different. And so not only do we lose these finger-like projections, but we also lose the crypt structure at the bottom, that's sort of the pit structure at the bottom of these finger-like projections. And this is because these cells have been killed and they're simply not able to repopulate the intestinal lining. Now, we're actually able to measure this quantitatively. So using a blood test, we can measure levels of plasma citrulline in the blood of people undergoing high-dose melphalan treatment. And we can see that we get this really very predictable decrease in citrulline that tends to begin recovering from around day 15 to 20 after stem cell transplantation. And this is a really important thing because in those slides I just showed you, they were actually collected from an animal model of chemotherapy-induced gut injury. But of course, in people, it's very difficult to get biopsies and samples of the gut. So this has been a really important way in which we can monitor people's gut injury in a relatively non-invasive manner. Now, when we think about the impact of this gut injury on people, we know that the average adult gut is around 30 meters squared, maybe a tennis court or two, depending on the person. And it's this large surface area that is absolutely critical for all of those important functions of the gastrointestinal tract. So the absorption of nutrients, the absorption of water. It's also a really important barrier structure because, of course, everything we put in our mouth is relatively dirty. It might have toxins or other pathogens in it. And so the barrier properties of the gut are really important as well. And so when we have this damage in the lining of the gut, we see a 50% reduction in the surface area of the gastrointestinal tract. And so understandably, we can appreciate that we have lower nutrient absorption, we have lower water absorption, and we have reduced barrier function. Now, symptomatically, I should say, this is why we see malnutrition being quite common in people undergoing chemotherapy. We see diarrhea, and we also see things like infection. And this is because we have bacteria moving from the inside of the gut across this damaged barrier and into the blood. However, what we're increasingly recognizing for some of these myeloma drugs is that there are many more mechanisms beyond this sort of indiscriminate damage to rapidly dividing cells. And this is particularly the case for some of the other drugs that are commonly used in the context of myeloma, like bortezomib or Velcade. And this drug is somewhat different, I suppose, to chemotherapy drugs because it doesn't necessarily work by damaging rapidly dividing cells. It works by inhibiting proteasomes. And what we know is that nerves, so these are the cells within the body that create the action potentials that allow different body systems to move and operate. These nerves are very susceptible to damage by bortezomib. Now, in the gastrointestinal tract, these nerves are what push food from our mouth through our esophagus, through our intestines, and hopefully, eventually, out the other end. And so what we're able to show using some models of bortezomib is that we see that food simply is not moving down the gastrointestinal tract. And we're able to measure this by measuring the stomach weights of mice that have been treated with the drug bortezomib. And so essentially what we're seeing here is I guess almost a paralysis of the gastrointestinal tract. And this is the mechanism by which we believe constipation is being caused by some of these drugs. Now, when we think about the gut microbiome, these bacteria in the gut, what we know is that the human gut is home to trillions of bacteria. And in the adult gut, it's around 2 kilos that are found within the large colon or the large bowel. I mean, 2 kilos is a huge amount of bacteria. And in fact, it's now considered that if you compare the amount of bacterial genes or DNA compared to human DNA, there's actually more bacterial DNA on the human body than there are human DNA. And so in many ways, we are more bug than human. And so it shouldn't be surprising that these bacteria are absolutely critical to our health, and they support a healthy immune system, they support brain function and metabolic activity, so hormone production. In particular, the hormone serotonin, which is our feel-good hormone. This is directly produced by interactions between bacteria and the cells lining our gut. And In fact, if you take an animal and you completely eradicate all bacteria from that animal, it's completely unable to survive unless it's kept within a really confined environment where there are no exposures to any pathogens or anything dangerous, because they simply do not have any strong or robust immune responses to be able to deal with these challenges. Now, what we know about the bacteria that live in our gut is that they tend to rely on two very important things. That seem to be working. There we go. Is a warm and comfortable home. And in this context, that is the intestinal lining. And the second thing that they rely on are regular meals, just like us. And this is typically provided to these bacteria by the fiber that we, the host, ingest. And so if you think about those two mechanisms that I described over the last few slides, we can appreciate that myeloma drugs impair both of these things. So they damage the lining of the gut, destroying the home that the bacteria really very much rely on to survive. And the food that we ingest is simply not making its way down to the gut to support these bacteria. And so essentially what we start to see in the microbiome of people undergoing treatment for myeloma is that it moves from being this beautiful, diverse, lush ecosystem of bacteria into something that looks quite damaged, quite depleted, and generally much less diverse. I mean, this is an extreme example here, this desert. It's absolutely not as obliterated as this suggests, but I guess you could maybe compare it to a slightly less intense rainforest. And so we're able to measure this, and this is called microbial diversity. We're able to take fecal samples from people undergoing myeloma. This is some data from people undergoing stem cell transplantation for myeloma. And we're able to look at the number and the relative balance of different bacteria in these samples. And we can see that over time, so from day 0 to day 30 after transplant, there is a significant decrease in the diversity of the microbiome. This does luckily recover. This late phase shown in the red is beyond 30 days, but to be honest, most of these samples were collected around 90 days after transplant. So it does take quite a long time for the gut microbiome to recover. We're also able to look at the number and types of bacteria that inhabit people's gut. So this is again that same cohort of people, and each vertical bar represents a sample that has been taken. On the left-hand side, we've got individual samples, individual patient samples collected over this time course. And each of the colors represents a different family of bacteria. Now, what I've shown here graphically is that bacteria shown in greens and blues, they're typically the bacteria that we would consider good bacteria. And the ones shown in darker red and pinks, they're typically these bacteria that we would consider bad. These are pathogenic bacteria that tend not to be so good inside the gut. And we can see that over time, as that microbiome becomes less diverse, we get a domination of these bad bacteria. And so this is what we call dysbiosis, which is essentially a term used to describe the balance of good and bad bacteria in the gut. Now, increasingly, what we're starting to understand is that these dysbiotic changes in the gut microbiome actually predict a range of unwanted consequences. So we can appreciate immediately that they impact gut health, typically having a domination of these bad bacteria in the gut will lead to things like diarrhea and maybe some abdominal pain. But we also know that it is the biggest predictor of sepsis or bloodstream infection in people undergoing stem cell transplantation. So this image on the left-hand side here shows it's a swab of blood that we've collected, and it shows that there are E. coli present in this blood that have directly translocated, so they've moved from the lumen of the gut into the blood. And so I guess this somewhat challenges what we have thought for a very long time. It's always been considered that many of the infections we see in people who are immunocompromised come from some external environment, perhaps hospital services, infected catheters or central lines. But in fact, they're coming from the bacteria that are naturally present within our gut. But these bacteria or these bad bacteria are typically kept at very low levels. So as a result, we see an increase in the duration of hospitalization in people that have worse microbial damage after treatment. We also see increases in the nutritional requirements for these patients, so an increased need for parenteral nutrition. And increasingly, there is data to suggest that the worse and the longer the duration of dysbiotic changes in the gut microbiome, the higher the risk of disease progression and even relapse. And so as a result, I would say that there has been this really intense and enthusiastic investigation of how we can better support gut health to avoid these unwanted consequences. And when we think about how we can support the microbiome, there are a number of different ways. Firstly, there are what we call prebiotics. These are typically dietary strategies that are used to support gut bacteria by providing the energy input that they need. And secondly, there are probiotics, and these are often referred to as bugs as drugs. So this is when we directly provide the live bacteria, which are usually provided in a capsule, and these can be used to then colonize the gut. Now, in terms of probiotics, there has been a lot of hype, but not necessarily a lot of evidence. I think the last estimate I saw was that the probiotic market is something around $95 billion US. It's a huge market. This has been an area that has grown very, very quickly. And certainly there is some emerging evidence, experimental evidence, to suggest that probiotics slow the progression of smoldering myeloma into active disease, and some evidence to show that they can reduce the symptom burden of treatment. However, it has been extremely challenging to draw robust conclusions because there are many different types of probiotic formulations available in the market, and each study seems to use something different. And so it has been really, really difficult to draw consistent conclusions across these studies. And while I don't have time to go into it today, I would absolutely recommend people have a look at this paper here. It does show and go through all the studies that have been conducted using microbial-based interventions, whether that be probiotics or prebiotics. But I think there's this statement or this perspective that probiotics are safe. You know, they're generally not considered a medication, they're good bacteria, so why not just take them anyway? And I think what's really important to emphasize is that this is somewhat counterintuitive, but actually probiotics can reduce the diversity of the gut microbiome. So if we think back to that rainforest image, when we give just a couple of types of bacteria, which most probiotic formulations are, you tend to dominate that microbial ecosystem or that rainforest with just one type of bacteria or one type of tree. And in fact, there has been a really important study that came out a couple of years ago that showed that probiotics given to people who have just had a course of antibiotics can actually slow the recovery of the gastrointestinal microbiome. Interestingly, this study also compared the use of fecal transplant. This is a method where we collect fecal samples, we process it, and we give it to the patient either by a colonoscopy or by an enema or by capsules. And this study was able to show that if you gave a fecal transplant after antibiotics, we saw a very rapid recovery in the microbiome. And so for this reason, fecal transplants really have become quite an area of intense interest, particularly in stem cell transplant recipients. However, the majority of evidence is absolutely focused on the allo transplant setting. And I think another really important factor to consider in this setting is that whilst there has been a lot of interest in fecal transplants and also probiotics, and certainly we've started to see these strategies used as a treatment strategy for things like graft versus host disease in the allo transplant setting, these have typically only been applied as a very, very last resort. And this is because there's a lot of fears around the risks of providing bacterial interventions in people who are immunocompromised. I mean, for a long time, we've even prevented people from eating food that may have some degree of bacterial presence on it. So it really has remained quite a controversial area. However, there is a really nice study that was, or a review, I should say, of the literature that was published recently that evaluated the safety of using probiotics in people undergoing stem cell transplant. And they were able to conclude that the evidence really does not suggest there is any additional risk of using these products in a patient who is immunocompromised. However, they did absolutely recognize that there haven't been direct comparisons. Most of the evidence has been collected retrospectively, so by looking back at studies or clinical practice. But if we think about the intestinal environment, and if we think back to that idea that actually intestinal damage or toxicity and the microbiome is predictive of infection, we can begin to actually put forward a consensus or a perspective that providing good bacteria would actually decrease the risk of infection. And so this is some data coming out of the Netherlands that actually shows the damage to the mucosal lining in the gut is a bigger predictor of infection risk compared to neutropenia. And so from this, we would suggest that actually supporting good bacteria, providing bacterial interventions in this time of severe immunosuppression may be a protective mechanism to reduce infection risk. It feels very counterintuitive, it feels risky, but scientifically it really does make sense. So I think that this is really going to be an area of intense investigation over the next little while, is how we can deliver and support the microbiome in a safe and effective way in an immunocompromised patient. Now, when we think about how we can do that, we can certainly begin to appreciate the complexities of delivering a fecal transplant to someone who may be very immunocompromised. This is certainly a challenging prospect. Not only are they immunocompromised, but they're also generally quite unwell. And so providing things like an enema or a colonoscopy, potentially even capsules, may be extremely challenging to achieve. And so something that we've been looking at is how we can indirectly support the gut microbiome by supporting the intestinal lining. The lining of the gut that I mentioned previously was damaged by chemotherapy. This damages the home of the microbiome and therefore leads to these microbial changes. And so I collaborated with some colleagues over in the Netherlands to conduct a study using the drug anakinra, which is an anti-inflammatory agent, which has been shown to be able to support the lining of the gut in other conditions similar to those seen after chemotherapy. And we were able to show that when we used anakinra, when we supported the lining of the gut, we were able to reduce the expansion or the overgrowth of some of these bad bacteria. So this little section here on the graph is very high in the chemotherapy only treated animals. However, when we compared it to that of our anakinra treated animals, there was a significant reduction in this overgrowth of bad bacteria. We then also were able to show that this reduced this very intense peak in body temperature. So this is fever, indicating that there has been an infectious event occurring. And so for us, this was really promising data to suggest that we might be able to support gut health and these gut bacteria indirectly by supporting the lining of the gut and making sure that that is not damaged as severely as it would be under normal circumstances. So I think I'm getting pretty close to the end of my presentation now. So I just wanted to emphasize that this is some really exciting research that we did conduct, and it has now actually transitioned into a phase 2 clinical trial, which is currently almost finished in the Netherlands. And we're really excited to see that hopefully this can be a new strategy to minimize the infectious and unwanted consequences of gut dysbiosis or gut damage in people undergoing stem cell transplantation for myeloma. So with that said, the take-home messages from my presentation today are that the drugs used to treat myeloma really do adversely affect gut function by damaging the lining of the gut and also the nerves that control the motility of food throughout the gut. These changes lead to disruptions in the gut microbiome, which, as I explained, can increase the risk of adverse events and worsen treatment outcomes. And we're really working very hard to actively try and design better ways to support gut health that are not only effective, but also really are truly feasible to implement. So these might be things like fecal transplant, which I am also working on at the moment, but also indirect methods like anakinra. I would like to finish up by thanking all the members of my lab and also my collaborators in the Netherlands, as well as the haematology department at the Royal Adelaide Hospital. And of course, the generous donors and funders, study participants, patient advocates, and their families. And if you're interested in connecting, please feel free to reach out if you have any questions or project ideas. We're always really happy and excited to learn from the lived experiences of people and hopefully together work to improve the outcomes for myeloma. So thank you very much.
Thank you, Hannah, for giving us such a great talk that was so interactive, so informative, and you explained the concepts of the gut biome so well. I think we do have a few questions from the live chat. The first one is quinoa and cacao, are they useful in improving gut biome at all?
So kefir is a form of probiotic. Yep. So this is a type of food delivered probiotic, which I didn't actually mention in my presentation today. There are absolutely a number of foods that have probiotics, so yogurts as well, and kombucha and all of that. Again, it comes down to this fact that different studies have adopted very different strategies. So, it might be someone taking one spoonful of kefir, one spoonful of yogurt, and a kombucha drink. So there are inconsistent reports of positive and not negative, but not positive effects. So it's been a really difficult field to synthesize the evidence from. Certainly there is some supportive evidence, but across the board, the general take-home message is that if you eat a healthy diet and do not have any gut diseases, typically these interventions don't have any effect. If there are reasons why your gut environment is damaged, then these can have some effects, but it is really important that those, if you are trying any of those interventions at home, that you do mention them to your doctor, because there are examples where they can worsen treatment outcomes.
Thank you for answering that. We have another question. So for people who are on lenalidomide who have chronic diarrhea, how would that be best managed?
It's a really good question, and actually a project that I'm about to start with a colleague of mine at the Royal Adelaide Hospital, Angie Young. So we're essentially at the moment, there is very limited understanding as to why lenalidomide even causes diarrhea. And so without that insight, it's really difficult to provide some practical solutions as to how it can be best managed. And unfortunately, that's why most doctors are really only able to provide very superficial interventions. I know that there is some evidence to show that bile acid sequestrants, so these are drugs that bind up all the bile in your intestinal tract, that these have some efficacy. And this is because lenalidomide is thought to potentially damage the part of the intestine that absorbs these bile acids. And so when they're not absorbed, they reach the colon, the large intestine, and they can be very damaging to the large intestine. So I think that's probably where the best evidence is at the moment, is for bile acid sequestrants, but it seems that different doctors have different preferences on that as a therapy. So I would absolutely encourage you to talk to your doctor about bile acid sequestrants and get their perspective on it.
Thank you so much, Hannah. That's all the questions we have. And thank you again for such a great explanation and just such a great informative talk.
Thank you very much. Thanks.
Thank you. So I would like to introduce a little bit our next speaker. She is Jessica, and I'll just give you a little bit of an intro with her. So, Dr. Jessica Hafner is a consultant neurologist at the Central Adelaide Local Health Network, and she has a special interest in peripheral neuropathy. She completed a neurophysiology fellowship at the Royal North Shore Hospital and currently runs the Complex Neuropathy Clinic at the Queen Elizabeth Hospital. She is currently the primary supervisor for the Central Adelaide Local Health Network Neurophysiology Fellowship, and she has a very keen interest in teaching and quality assurance. So let me hand you over to her.
Thank you very much. I'll just get started. We've only got half an hour and I've got a little bit to get through. So I've been asked to talk to you today about myeloma-related neuropathy, particularly with a focus on what you might be able to do about it. So firstly, what is neuropathy? What are the types? Why does it happen in people with myeloma, and how is it diagnosed? And then I'd like to focus most of our time talking about how we can prevent it, how we can manage symptoms, and then just touch on what research is happening in this space. So just very sort of basically, peripheral nerves are a part of our body that carry electrical signals to and from the brain and or the spinal cord to give us information about sensation, to help us move our muscles, and then also to control other bodily functions, such as Hannah was saying, our gastrointestinal muscles, and also our bladder muscles, blood pressure, heart rate, and sweating are also controlled by nerves. And peripheral neuropathy just is a term that's sort of all-encompassing and refers to any condition in which those nerves are not working properly. And that can be in various forms. So you could have just a sensory neuropathy affecting the nerves that conduct messages about sensation, or a combination of sensory, motor on its own, or sometimes involvement of those autonomic nerves that control bodily functions. And I just thought I'd also mention the terms large fibres and small fibres, because I'll refer to those later in the talk. So large fibres are named so because they're physically larger, they're thicker, and they carry information about touch, joint position and vibration. And small fibres are smaller, obviously, and they carry information about pain and temperature. And that's just relevant because sometimes in myeloma, people can have one type of fibre affected more than another. So in terms of the symptoms, it depends which of those nerve types is affected, what the symptoms might be. But just very briefly, people who have small fibre neuropathy will get symptoms like numbness, pain, and nerve pain is classically sort of a burning or even like an electrical type pain, very sharp, often very painful, and also can get tingling and hypersensitive skin that sort of feels painful when you touch it, and the type of touch wouldn't normally be painful. People who have involvement of their autonomic nerves have symptoms associated with some of those functions not working properly. So getting dizzy when they stand up, having trouble with constipation or diarrhea, trouble with emptying the bladder, dry eyes, dry mouth, impotence in men, and also feeling full or even sometimes vomiting after meals because their stomach's not emptying normally. And this can then lead to weight loss. People don't tend to complain of a lack of sweating, but it's something that as a doctor interested in neuropathy, I often inquire specifically about. And then large fibre symptoms, again, numbness and particularly that sort of difficulty with joint position. That's how our sort of legs tell us which way we're standing. So when that doesn't work, we tend to lose balance. And then if it's affecting the nerves to the muscles, we can get muscle cramps or even weakness or wasting of the muscles. And then often, particularly in amyloidosis, which we're not talking about specifically today, but often is related, carpal tunnel syndrome is very common, and that involves one of the nerves in the wrist being sort of damaged particularly, which can cause hand symptoms. And then also in amyloidosis, it's possible to also get muscle symptoms unrelated to nerves. And the reasons that people with myeloma and amyloidosis get neuropathy is actually quite complicated. It's not a simple single problem. It can be from the paraprotein itself in myeloma and in AL amyloid, which can be directly toxic to the nerves. Also, if you've got very high levels of paraprotein, your blood can be sort of sticky or viscous and not flow through blood vessels as easily, and therefore your nerves don't get as much blood supply and oxygen as they need, and that can cause damage. Myeloma bone disease, if you've got a nerve sitting very close to a bone and then you get a bony lesion right adjacent to it, it can expand and sort of stretch or crush the nerve. Having a form of cancer in general, and myeloma is no exception, can cause weight loss, which we actually rely on some of our adipose tissue, our fatty tissue, to protect and pad our nerves from compression. So when we lose a lot of weight, our nerves are more vulnerable to being crushed or compressed. And also, as Hannah was talking about a lot, having nutrient deficiencies in cancer and cancer treatment can also be a big problem for nerves, which rely on quite a lot of different nutrients to function well. Also can be more vulnerable to certain infections, such as the shingles virus, which sort of tends to get into and damage nerves. And then also the treatments that are used in myeloma and in AL amyloid can also increase the risk of neuropathy, particularly bortezomib, which can be directly toxic to the nerves, or less commonly actually cause the immune system to sort of malfunction in a way that causes it to attack the nerves. Thalidomide can also cause neuropathy. We don't really understand why that happens at this stage. And then vincristine, which is not used very commonly in myeloma, is well known to be quite toxic to nerves. And then, of course, the common things that cause neuropathy in the wider population can also affect people with myeloma. So things such as diabetes, excessive alcohol consumption, and vitamin deficiencies, particularly B12, can contribute. So in terms of what you will be looking for if we're examining you to work out if you've got a neuropathy, we'll be looking for those things I mentioned. So is there loss of sensation? Is there a drop in blood pressure? Is there difficulty with feeling joint position, losing reflexes, and looking at muscle wasting? They're the sorts of things I look at when I'm trying to diagnose a neuropathy. And then we also might do some tests and nerve conduction studies, which some of you may be familiar with, where we put sort of electrical pulses through the nerves and measure how well they transmit those electrical pulses. That's a very useful test for working out where the problem is, what type of problem it is, and how severe it is. In people who are having symptoms of autonomic neuropathy, so difficulty with, feeling full or feeling dizzy when standing, there are some specific specialized tests to look at those. So gastric emptying studies and urodynamic studies, which look at how the bladder is emptying, and tilt table testing, which is a sort of testing whether your blood pressure and heart rate do what they're meant to do when you get tipped from lying up to standing. And we also obviously will look for things that we could treat, so things that could be causing neuropathy or worsening neuropathy apart from myeloma and treatment. So testing for diabetes, testing for B12 deficiencies. And then in some situations, if the neuropathy looks a bit strange or unusual or not what we're expecting, we might do some additional testing, such as testing spinal fluid or even urine samples, to look for a less common cause of a neuropathy, which might require a different specific treatment. Sometimes, but not always, and actually not even often, if we're very concerned that there's a problem with the nerves that we haven't really got to the bottom of, we might take a biopsy of the nerve, where we look at a piece of nerve under a microscope. It's actually the only way that we can definitively confirm that someone has an amyloidosis-related neuropathy, but it's a procedure that's not without risks. It can cause obviously, it will cause a patch of numbness because you remove the nerve, and some people will also get post-operative pain afterwards. So we don't do it unless we think there's really no other way to work this out. And if somebody has known myeloma or known AL amyloidosis, we generally wouldn't need to go to the nerve to confirm the diagnosis. So does everybody who has a neuropathy need to see a neurologist? Well, it's a tricky question, and I think it really depends on access. And I'm not sure where everyone I'm speaking to today is based, but here in South Australia, we have a very substantial sort of supply and demand mismatch with our neurologists, and there's a lot of people who would benefit from seeing a neurologist, but not enough people to see them. And so unfortunately in South Australia, we're really restricted to seeing people where we think we're going to add the most value to their care. And if there are other options, then we try to support general practitioners and physiotherapists to care for our patients with neuropathy without necessarily needing to see every single person. But certainly if people are having difficulty walking or difficulty with their balance or muscle weakness, or their neuropathy is getting worse quite quickly, those are the people that definitely need to come and see us. And the reasons for that is that we can confirm what the diagnosis is, make sure that we're not missing something that needs a different strategy for treatment, making sure we pick up any neuropathies that are potentially treatable. And then also we like to help to guide with any symptomatic treatment. And often we sort of collaborate and work with general practitioners, hematologists, oncologists, pain specialists, and of course our allied health colleagues, physiotherapy, occupational therapy, and dietitians to sort of support patients with neuropathy going through that, even when we can't necessarily fix the neuropathy, for want of a better term. So what can we do then to prevent treatment-related neuropathy? So this is the neuropathy that occurs due to the treatments that are used to treat myeloma. And this is really tricky sometimes because it's a little bit of a catch-22 in that to prevent treatment-related neuropathy, obviously if you didn't use the treatment, you wouldn't get it, but the treatment is needed for the myeloma, which has its own issues, of course, as you all know. So the things that we can do is promoting nerve health. So if a person with neuropathy also has diabetes, then a really good focus on good control of their blood sugars goes a long way to supporting their nerve health. Avoidance of alcohol, and in most situations, this doesn't need to be complete abstinence, but certainly alcohol is toxic to nerves, and if it's being consumed excessively can contribute significantly to worsening neuropathy. Smoking cessation has lots of advantages, and if we're wanting to make sure our nerves have good blood supply, then stopping smoking is essential. We want to make sure we diagnose and treat any vitamin deficiencies that might exist, and maintaining a healthy and nutritious diet will ensure that those deficiencies don't recur. Some of the medications that are used in myeloma are sort of non-negotiable, and obviously if people have a severe neuropathy, we want to have a look and say, is there anything that we can change here that will improve the chances of this neuropathy not progressing to be something that's significantly disabling. And so sometimes hematologists will review doses and change regimens to reduce the risk of that severe progression. But obviously that needs to be weighed against the risks of potentially less effective myeloma treatment as well. And that's quite often quite a complex discussion that needs multiple people kind of working together, including, most importantly, the person receiving the treatment, to weigh up the pros and cons and what's important to them. There is a fair bit of research out there around, is there something we can do to prevent treatment-related neuropathy? So when we know we're going to be giving somebody a medication that could cause a neuropathy, is there anything we can do to reduce the damage. And it's a bit unclear. People have looked at various sort of therapies about temperature control and compression and various medications and agents. And at this stage, there's not anything that is clearly in favour of doing those things related to myeloma medications that are used. Some of the other chemotherapy agents used in other conditions, there seems to be some things that we can do to reduce the risk of neuropathy, but in the myeloma space at the moment, nothing's yet been proven and in routine practice. So then if we do have neuropathy, what can we do about the symptoms and how can we minimise the impact that it has on our lives? The biggest issue, I think, often is neuropathic pain, and this is a really tricky space because a lot of the medications that are used to treat neuropathic pain have lots of side effects and also aren't sort of excellent in their efficacy. So I always talk to my patients about non-medication measures, and pain's really complicated. It's not just a biological process. It's so closely intertwined with our own sort of social and psychological and physical well-being. And the way our brain receives and interprets messages about pain is something that we're still growing our understanding of it. But there's lots of really good scientific evidence now that looking after those sort of social and psychological and physical well-being aspects goes a really long way to reducing the impact that pain has on our lives and the disability that pain causes. So I really encourage patients to think about those things and actively try to address them. And this is things like staying active with your family and friends and engaging in activities that give you meaning and things you're passionate about, practicing relaxation and mindfulness and doing attentional techniques, so diverting attention away from pain will reduce the amount of sort of volume that you're I often think about it like turning a knob on a radio, and we want to turn down the volume on the pain signals coming into the brain, and diverting your attention away from it. And that's obviously not very easy when you're in severe pain, but it's something that you, with expert input, you can practice and master, can sort of go a long way to turning down the volume on some of that pain. Formal psychological assessments can be helpful. They're not for everybody, but particularly where there seems to be a real connection between pain causing low mood or low mood in turning the volume up on pain, then formal psychological input can be really valuable. Physical activity, every doctor you have talked to will tell you to exercise more, and I think that's very easy to say and often very hard to do, particularly when you're in pain and particularly when you've got fatigue and you're experiencing side effects from chemo. Getting out there and doing exercise is often doesn't feel like a very realistic goal. So I always talk to people about trying to find something that they enjoy doing. It doesn't have to be a particular type of exercise. It has to be something you enjoy, and because that's the thing that's going to predict whether you're actually able to do it and actually able to get there. Doing things with friends, just starting slow, starting at a very achievable level, not trying to hit the gym suddenly when you haven't been there for 25 years. Just gentle walking, getting outside, getting some sunshine on your face, that sort of thing, I really encourage people to do. TENS machines can be helpful. Again, not for everyone. Some people love them, some people hate them, but this is like a electrical nerve stimulator that you stick pads on your skin, and physiotherapists can often teach you how to use those and encourage and sort of show you how to best apply them. And then acupuncture can also be helpful, but in the setting of myeloma and chemotherapy, it's really important you discuss it with your doctor to make sure it's safe for you, because if your blood is not very good at clotting, then getting a needle stuck in your skin might actually cause you more of a problem. So definitely discuss that one before you go down that path. Medications, I think it's the sort of key things I would highlight from this slide, there's a lot of information here, is that using paracetamol, unless there's a reason that you can't use it, Even though on its own it probably won't make a huge amount of difference to your pain, if at all, there's lots of evidence that by using regular paracetamol, the other medications you use will work better, and therefore you can use them at lower doses and therefore have less side effects from them. So I encourage everybody to use recommended doses of paracetamol, obviously in discussion with their doctor, if they've got a chronic pain problem. The other medications your doctors might use to manage your neuropathic pain include medications from various sort of groups. So some of them are antidepressant medications, some of them are anti-seizure medications, and they all work by acting on the chemical signaling from nerves to try and turn down some of those pain messages. I think with all of these medications, they do have side effects, and some people find that that is the reason they can't get good control. So I always recommend that people start on very low doses and increase them very slowly. And this helps with side effects not being as problematic, although it does, of course, make it a longer and slower road before you get the benefit. And so patience is required. It can take a few weeks before you really know whether each dose change has made a difference to your pain levels. And I also encourage people to really have some objective measures. When we're in pain, it's very easy to only be able to remember what, like if I've had a really bad day of pain the day before I come and see my doctor, I'm going to say, oh, my pain's terrible, even if I haven't necessarily had a lot of pain in the last month, because pain is very all-consuming and really occupies a big space in our brain. And so I like to keep diaries and sort of set goals and track things, because if you have one really bad pain day, but that's the only one in 6 weeks, then that's better than having bad pain, 3 or 4 times a week. So setting those things down on paper can help us kind of see where we're making gains and see what's working and what's not. And then also it's worth talking to your doctors about topical treatments, such as capsaicin, which is like from a chili plant, and lignocaine, which is a local anesthetic, and these things can help with localized pain and have sort of less in the way of side effects, because they're not going into your bloodstream in the same sort of doses that a tablet will. The most common reasons I see that people don't have success with medications with neuropathy pain is sort of not using paracetamol, starting with really high doses and then getting side effects and stopping it, or starting on really low doses and then never increasing them, and so not taking enough. Taking it for 3 days and saying it didn't do anything, and stopping and not giving it a chance to sort of give that 2 to 4 weeks lead in that you need. And then also sometimes, and often not because the person taking the medication has done the wrong thing, but because they've been given some sort of unusual advice. Sometimes some of these medications get prescribed as sort of as needed. So, and they don't work like that, because they change sort of nerve signaling over time. And so taking a sort of an antidepressant or an anti-seizure medication for neuropathic pain just kind of when you get your pain is not going to, they don't work like a Panadol where half an hour later you get relief. They need to be taken regularly to have their intended benefit. I think it's also, and this is a bit of a sort of disheartening thing to put on here, but sometimes pain cannot be completely suppressed, and that can be very frustrating, of course, but sometimes that sort of expectation that a medication will make the pain go away completely, I think, can set people up to keep trying new things, searching for that thing, rather than sort of benefiting from an improvement in pain and having some more realistic expectations. And they're people who I really would encourage to look at some of those non-medication based pain strategies, because the medications we're not there yet, unfortunately, in terms of having sort of a silver bullet for neuropathic pain in terms of a medication. So then in terms of muscle cramps, this is another really common symptom that my neuropathy patients talk to me about, and it's something that's not really well dealt with in the literature, in the there's not a lot of research around what to do about nerve-related muscle cramps. And so the advice is really restricted to some very sort of generic statements, but these are things that are helpful. So making sure you're adequately hydrated, obviously not, I'm not suggesting anyone goes out and drinks 8 litres of water a day, but just making sure that you're not thirsty and you are keeping up adequate water. Reducing or avoiding alcohol can also help, as that sometimes is related to dehydration and also muscle cramps. Doing gentle stretches, particularly before bed. Often people get muscle cramps at night time in bed, and it disrupts sleep and can be very painful. So doing stretches before getting into bed, particularly stretching the muscles that tend to cramp. And then also massaging during a cramp can help, and stretching it gently can help relieve it. Sometimes a nice warm bath or a warm shower before bed, and also making sure you're not cold in your bed, can help muscles to not cramp as often. People can sometimes you'll have people recommend that you take magnesium or that you take quinine or something like this for cramps, because they're sort of people think that they help with cramps. I think it's important to be cautious about these medications, particularly when you've got other significant health issues going on, such as myeloma. So if you had low magnesium levels on a blood test, then certainly a magnesium replacement is very reasonable. But if you're taking it and you don't need it, it can actually cause side effects, including sort of getting low blood pressure, feeling nauseous or headachy, or even diarrhea, which given that we're already concerned about nutritional deficiencies, we're really not wanting to add to that. Quinine is not recommended. It can cause low platelet counts, which many of you know are the part of your blood cells that are involved in clotting, and very important. And so it can be very dangerous to use quinine, particularly in the setting of having chemotherapy for a blood cancer. So there are some other medications that people will sometimes try for cramps. None of them have very good scientific evidence behind them, but I think if you are having a lot of trouble with cramp, it's worth speaking to your doctor about what the options are, and they can talk you through the risks and benefits of those. Again, another busy slide, managing autonomic neuropathy. It depends on what the symptom is that is bothering you, but I've just got some of the sort of highlights here. So diarrhea, it's the first thing when someone tells me that they've got diarrhea and I know that they've got an autonomic neuropathy, I want to make sure that it is truly due to the gut sort of moving too quickly, and not due to probably a more common problem, which is someone who's very severely constipated and then gets loose stools, because that's the only sort of stool consistency that can get around the stuff that's all backed up. So a bit of a graphic description there, but it's what we call overflow diarrhea, and it's loose stools sort of coming around stuff that's all stuck in there. And actually the treatment for that type of loose stool is to actually use laxatives, which can be a bit counterintuitive. So careful sort of history taking and trying to work out what the sequence of events has been can help work out which is which. Sometimes we need to do even an abdominal x-ray to just have a look and see if we can see if the bowel is all backed up. So either increasing fibre, or even taking some medications to slow down the gut if we think it's truly diarrhea, or laxatives if it's constipation. If the stomach isn't emptying properly, then some sort of strategies like just eating small meals, and small meals more frequently, rather than having a really big meal that then fills the stomach up and makes you feel awful and vomit. Avoiding sort of things that are very high fibre or very high fat, which are kind of a little bit trickier for you to digest. And then speaking to a dietitian can be very helpful, and certainly is recommended if you're experiencing weight loss in the setting of this. Sometimes medications can sort of stimulate the stomach to empty more quickly, and so those can be useful. Difficulty with bladder can be tricky, because the most common type of bladder problem is a bladder that isn't very good at emptying, and so sort of retains urine and is therefore more prone to infection. And so there are things like urinating on a schedule, so not waiting till you feel the need to go, but just going sort of regularly to try and avoid that sort of urine building up and becoming a place where bacteria can live, and in some situations, self-catheterization. I've just got a message that I'm going over time, so I will move on. Apologies. Autonomic neuropathy with low blood pressure, there's a few things there, and I'm very happy if people want copies of these slides later to have a look at, they can do that. But it's around trying to increase your salt and particularly thinking about compression stockings or tilting the bed up. And in terms of looking after your feet and your hands, physiotherapy and podiatry are the really big key points here, and making sure that we look after numb feet and don't let them get injured when we can't feel it. I apologise for taking too long to talk. Vitamin supplements I really only recommend if you have a confirmed deficiency, and making sure you're not accidentally taking too much B6, which is a common ingredient in many multivitamins, and it's very easy to overdo, and that can be harmful to nerves as well. If you're taking any additional sort of herbs and supplements, then certainly talk to your doctors about it, particularly if you're on active chemotherapy, because these are often used because they actually do something, and so they can interact with your chemo and make it either less effective or more toxic, and it's really important that you discuss that with your doctor. And so medicinal cannabis, that's another one that commonly comes up. Commonly people will ask me about this because they hear on the grapevine that this is really good for nerve pain, and they want to know whether this is suitable for them. I think it's certainly worth looking at, and there is some evidence that it is helpful in nerve pain and also in chemotherapy-related nausea and vomiting. But it's important to note that although there is evidence, it's not recommended as the first thing to try in those situations. And it's unfortunately not on the pharmaceutical benefit scheme for either of those indications. So there's a significant cost associated with it, which at the moment is about $100 a week. And it's important that I mention, and I mention this to all my patients who ask about it, that procuring cannabis products online from other sources can be harmful because they are not regulated. You don't actually know what is in them. They might not have anything in them, and it's not legal to import them. So just always something that I mention. If you are interested in learning more about medicinal cannabis for nerve pain, the NPS Medicine Wise website, which I've got the link here for that, is a really good resource, and you can find more information about that. All right, sorry, let's keep going. So in terms of preventing progression, if you already have developed neuropathy, and unfortunately nerves, although they can heal, they do it very, very slowly, and in the setting of ongoing active medical issues, they often don't do it particularly well. So what can you do to stop your nerves getting worse? So the main things really is making sure there's not some other factor going on. So diabetes is really the big one that you want to get good control of if it is in the picture, and B12 deficiency is the other really common one that it's important to check for. Avoiding other medications that can cause nerve damage if that's possible, and unfortunately the list of those medications includes some of the chemotherapy agents used in myeloma, as we've discussed. Maintaining a healthy, balanced diet is the best way of making sure that you're getting all of those nutrients and micronutrients in. And if that is difficult because of other issues, then talking to your doctor about the need for vitamin supplementation, and then reducing alcohol use as well. So as I said earlier, in the presence of neuropathy, we know that makes it worse. All right. In terms of current research at the moment in South Australia, which is where I am, there's nothing locally happening on the trials register that I can find. But there are a few things that are on the Australian New Zealand Clinical Trials Register, and this is a really great place to go looking if you're interested in finding out what is around and looking to participate in any clinical trials. If you go to this website, you can put in various search terms for particular medical conditions or particular groups of people and find trials that might be relevant to you, and they have the contact details there for the people running the trials. And I think in summary, I'll just summarize. I did go very quickly through some of those slides, sorry, because I panicked about my timing, but obviously very happy to answer any questions or revisit slides if people wanted me to. So we know that neuropathy is common in myeloma, and optimizing nerve health by managing the things that we can control, such as diabetes and alcohol, is important to minimize symptoms. In some cases, dose and treatment regimens may need to be adjusted, and that will be weighed against the risk of treating the myeloma less effectively. Treatment of symptoms, particularly the pain symptoms and the autonomic symptoms, can really improve the experience of living with neuropathy, even if the neuropathy itself can't be fixed. And that's using a combination of sort of lifestyle measures and also the non-drug pain management, as well as prescribed medications. Vitamins and supplements very frequently recommended by well-meaning friends and family for people who have neuropathy, but it's really important to make sure that you discuss those with your treating doctors, because some of them can accidentally harm you, such as B6 supplementation, which I see probably 1 or 2 cases a year of people who have B6 related neuropathy from accidentally taking too much. And medicinal cannabis can play a role in neuropathic pain management, but only really should be considered where other measures have failed. And as mentioned, more information on the MPS Medicine Wise website about that. So thank you, and I'm very happy to take questions or revisit any slides that I might have inadvertently skipped over quickly in my time-related panic. Sorry about that.
That was excellent, Jessica. Thank you so much. Such an informative session, and you actually answered some of the questions that had come through in your slides, particularly on the neuropathic pain and management there. So thank you for those tips. We did have one with an autonomic symptom question, in particular relating to lenalidomide and some night sweats that they're getting. Is there anything you could touch on there in terms of management or prevention?
Yeah, so night sweats is tricky because it's probably not just related to neuropathy per se, and is probably multifactorial. Generally for night sweats, and obviously it might be medication related, it could be even myeloma related, like there's lots of things that could be contributing. And the advice is pretty sort of basic stuff, but for night sweats, I usually just encourage people to, make sure they've got cotton sheets and cotton pajamas, make sure their room is cool, make sure they've got a fan on, and just trying to reduce their temperature. But unfortunately, there's not really any sort of medication or anything else specifically other than trying to keep cool. Unfortunately, in a night sweat, what will happen is it will happen even if you're cool, and then you'll be really cold because you're now covered in water. And so, yeah, it's a tricky one, but I don't have any very clever solution for that one, unfortunately. Other than avoiding synthetic fabric is the only thing I can suggest.
That's a great tip. Thanks so much. Encourage people to look back through the slides, and you can always send your questions through to us at nurses@myeloma.org.au. Thank you very much again for your time, Jessica. We really appreciate you being here. Thanks so much.
Thank you. Thanks.
So that does conclude our program for today. We hope you found this session and all of the seminars over the month informative and uplifting. A reminder that you are welcome to send through any questions via that nurses email, nurses@myeloma.org.au. We really do appreciate your feedback, so please ensure you complete the evaluation via the link below this video on the webpage. Before we say a final goodbye, we'd like to express our heartfelt thanks to all of our presenters today and throughout the month, and to the seminar organising team at Myeloma Australia, who have coordinated a fabulous program. Thanks also to all of our sponsors, and a very big thanks to Darren and the team at CVP for their hard work and their skill in production. Thanks again for joining us, and have a wonderful day. Bye now.